Hello, everybody. Welcome to this webinar. Let me go ahead and share my screen. We'll get started in a couple of minutes. So there are lots of people joining, and we'll give them some time to log on. Sometimes the GoToMeeting requires you to download a short, small application. So we'll give people a chance to do that. So just so you know, or just as a reminder, today's webinar is on interpreting RNA-Seq data in the genome browser. And it will be presented by David Roos, who we will give him the controls in a few minutes. In the meantime, while we are waiting, we'll conduct a direct poll. So go ahead and you can use your phone. You can scan this QR code or just type the URL at the top, which is http etc/XQ5. So you can start this and hold tight. I'll go ahead and start that online survey in a second. All right, so this is the same QR code. So I'll go ahead and start it. The link still shows right here at the top. So if you did not get the QR code, go ahead and copy that link. I will show the QR code in a second. I'll leave this poll running throughout the webinar. So if somebody else joins late, I'll show the QR code later on, and you'll still have a chance to enter your responses. It's pretty cool. It's like a race. You can see which database is winning. Cool. Yeah, and if for some reason you're not able to enter your responses in the using the QR code or using this direct poll system, you can go ahead and type it in the question session. I just saw that somebody responded saying that they use Pyroplasma DB and Vectorbase. So that's cool. The poll is a multi-select. Excellent. So we have about 87 of the participants voted. We have close to 200 attendees in this meeting. So continue to vote. I will show the QR code again a bit later, as I mentioned, and we'll show this image again as well. But you can type, if you type this URL in your browser, you'll get to the questions, or if you scan the QR code, which I will show it right here. So if you haven't done so yet, you can scan this. While you're doing that, I will show the results in a second as well, just so you know. But just so you know, in GoToWebinar, it looks like a lot of you already know this functionality, but there is a questions section that allows you to enter your questions or comments. So please do so throughout the webinar. So my name is Omar Harb. I'm part of the outreach team for the ViewPathDB project. I'm joined today by a number of outreach team members, including Evelina Bashenko, who's based in Liverpool, Sam Rund at Notre Dame in the US, Gloria Geraldo Calderon, who's in Colombia, and Suzanne Warnfels, who's at the University of Georgia, and I'm based at the University of Pennsylvania. And so we'll all be looking at your questions and answering them throughout the webinar. And if need be, we'll go ahead and interrupt David during his presentation if we feel like it's useful for us to hear the answer verbally, or for him to demonstrate something or repeat something. We go back to the presentation. Well, let's take a look at the results here. So we have many more participants showing or participating in the survey. So that's great. This is very useful information for us. Obviously, we keep track of this from webinar to webinar, and that gives us an indication of which databases people use. There's also a raise your hand option in webinar, GoToWebinar. I've noticed somebody raised their hand. So if you have a question, just go ahead and type it. I'm not going to try and unmute people independently because there's just too many people on this webinar to do so. So go ahead and type your question if you do have a question. And often people end up pressing that raise hand option by mistake. And so to avoid interruptions, just go ahead and type your questions if you have one. All right, so going back to the webinar. So one other thing before we get started, well, two other things. One is this webinar is being recorded. So don't worry if you miss something, or if you want to share this with colleagues, you can do so. After the webinar, we will make the results of the recording of the webinar available to you, and you will be able to share it with others or view it yourself. Also, this page here has a list of all of our webinars. You will get a link to this page in the post-webinar email anyway, but if you want, you can go ahead and copy it right now. And I can show you what this looks like quickly. So it's a list of upcoming webinars. You can see our next week, for example. It's a webinar that's going to focus on building search strategies. And so if you are ever curious how you can use ViewPathDB resources to really interrogate the underlying data, for example, asking a question like, find me all genes that are potential kinases that are also secreted, that are expressed at a particular developmental stage of your favorite parasite. And so those are the types of search strategies or in silico experiments that we will be running during that webinar and teaching you how to use. And then after that, there's a webinar on Galaxy. So using Galaxy workflows to analyze your own data, and then manual curation on Apollo. We will be adding a number of additional webinars probably by early next week. So of course, come back and visit this page frequently and to see these webinars. Past webinars are also listed on this page, including the recordings. Okay, so I think this is all I had for now. I'm going to turn over the controls to David Roos. David, I'm going to just make you the presenter, and you can go ahead and share your screen when you're ready.
All right, I believe you should be able to see my screen and hear me. Is that correct?
Yes, we can hear you. Mm-hmm.
Good morning, everyone, or good afternoon if you're in Europe, or I guess good tomorrow if you're calling in from Japan or New Zealand or someplace to the far east of me here in Philadelphia. Following on from the webinar which Omar ran yesterday on JBrowse as a means of being able to view various different types of information in the ViewPathDB database supporting various parasite resources, fungal genomes, and vector genomes, I wanted to spend today to give some insight into how to dive a little bit deeper into the resources that are available, the information that is available, the quality of the annotation, ways that annotation could be improved, and provide a view towards the future, in particular a webinar that we are expecting to run in a month's time or so, in which Dan Lawson will take you through the new Apollo browser platform, providing a means for you individually or as a group, as part of your research community, to improve the annotation online. And I will give a little bit of insight into what's highlighted at the top of the page I'm showing here, the Toxoplasma database, ToxoDB, highlighting the merged vector-based ViewPathDB resources, including support for all of those genomes. I do want to remind you that all of the materials that we run in the many workshops that the ViewPathDB project has been responsible for are available from the toolbar at the left of the live site for any ViewPathDB genome, including the most recent ViewPathDB workshops, for example, illustrating schedules, or if we look at the most recent, if we look at the most recent of these schedules, coming up slowly here, we look at the most recent of these meetings run at the University of Georgia, for example, there are links to all of the exercises we run in a week-long workshop, and clicking on the links from that schedule allows you to bring up a page with the various exercises, including exercises that we'll be discussing here today. For example, a link to a page on interpreting RNA-Seq data based on an older architecture of the database provides a great deal of information of various different kinds of browser views in the gbrowse browser that Omar referred to last week. This has now been replaced with the jbrowse browser, and the exercises will be modified in due course. But this gives an illustration of where we're going in today's hall, is a mechanism for being able to look for the various genes, which in this case I've configured to show in the middle of the page, to take a look at supporting evidence, including chromatin mark data, promoter transcriptional binding data, RNA-Seq data in support of these or refuting these various gene models, synthetic information, and so on. Today, I'm going to concentrate on a particular gene in ToxoDB, but the same principles apply for whatever genomes you might be interested in Giardia or trypanosomes or Aspergillus or coccidiomycoses or in vector genomes, as they're integrated into the same underlying architecture. And as I say, I will give some examples of that later on. Once again, let me also remind you that although we will spend most of our time today on a particular gene in the ToxoDB website, if you have particular genes or particular genomes or particular organisms or groups of taxa that you are interested in yourself or in your lab group, I encourage you to click on the Contact Us link, your one size fits all starting point for being able to solicit assistance from the outreach team, who is happy to run webinars or work with you personally, sharing screens to be able to make sure that your particular questions of interest are addressed. The gene we're looking at here, the purine phosphoribosyl transferase gene, is a gene that we'll be looking at for a couple of reasons. First, because it's a gene that I'm very familiar with, having worked on this over many years in a long series of publications, highlighting, for example, dating back to the first identification and cloning of this gene nearly 25 years ago now. This gene was, at the time, the first identified alternatively spliced gene in, I believe, any parasite system. And you can see that from classical molecular biological approaches, it was identified as having a couple of different polymorphisms, an exon skip polymorphism, in that this gene, which is annotated as having 1, 2, 3, 4, 5 exons and 4 introns, in fact has 1, 2, 3, 4, 5 introns here. And alternative splicing leads to the inclusion or exclusion of a coding exon here that encodes, I believe, 49 amino acids that have important functional characteristics associated with the two isoforms 1 and 2 of the HGPRT gene. This gene was also annotated as having an intron read-through variant that occurs only in isoform 2 and takes place in the non-coding sequence in the 5' UTR of this transcript. So we're going to spend our time today asking how well this data and annotation holds up in light of the vast wealth of new information that is available through these various databases. So I have gone to ToxoDB and navigated to this gene by running a search for phosphorybosyl transferase or the name HXGPRT or the gene ID, if you know that. And I come to a gene page which provides me a variety of alternative views of looking at this gene in the context of other related species, alignments with available sequence information, phenotypic data, polymorphism data, transcriptomic data, and all sorts of other information, as you know from previous webinars or your own efforts. As we scroll down the page, we begin to look at various browser views, and we can see straight off that notwithstanding a 25-year-old annotation indicating the publication indicating the alternatively spliced variants, only one of these variants is indicated in the annotation. In fact, this variant corresponds to neither of the two published variants, one of which had a read-through in the first intron, and the other of which had a skip of the third exon. We'll come back to the evidence for introns that is indicated right up front on the gene page and look at that in greater detail in the context of the JBrowse genome browser. We will not be looking today at the GBrowse browser, a product which is no longer supported and will ultimately be retired, but which is the basis of the current tutorial that you can access as I showed you earlier. If we jump down to look at additional transcript information, you'll see a summary of transcription data from the many RNA-seq transcription profiling studies that are represented here. And we can see, as is typical for many genes in many of the organisms that folks on this call are interested in, that the transcript abundance of this gene as measured in FPKM values, the number of reads per million reads from a transcript, varies over 2 to the 10th fold, over 1,000-fold from low, essentially no expression in toxoplasma oocysts to higher levels of expression in various different stages. If we click on this, we should be able to see that there are moderate levels of expression in the acutely lytic tachyzoite stage of toxoplasma parasites, lower levels of expression in the tissue cyst stages, and very high levels of expression in the epithelial stages of a cat intestine that are producing gametocytes, precisely when one might expect that a nucleoside metabolic enzyme like this would be highly expressed. All of those studies and more, based on microarray analyses, are represented down below, and we can dive more deeply into these as you've seen before in other webinars or are welcome to explore for yourself. We can examine, for example, that looking at the time series of an acutely lytic stage, we see roughly similar levels of expression. We could look at various different strains and observe the expression at different times here based on a microarray analysis, for example, which should tell us that consistent with the RNA-seq data summarized above, expression in oocysts is very low, in tachyzoites moderate, and in this particular experiment, which is taking a little bit of time to load, we'll see we see exactly consistent results here. Recall that this is displayed on a log scale. But the data set that showed us the greatest range of expression was this data set down below, representing the various stages of gametocyte differentiation, which now displayed on a linear scale, are vastly higher even than the moderate level in tachyzoite parasites. So let's go and take a look at what this expression profile looks like. We're going to turn on now a view of transcriptional expression data shown for this particular gene in the various life cycle stages here. And I'd like to dive in a little bit further to open up that view in the JBrowse genome browser and take a look at the transcriptional profiling coverage plots as summarized here in this screenshot that's embedded within the gene page.
David, while this is loading, there was a question that I thought might be useful for you to answer right now before moving on. And the question is from Stefan Vella, and it's what would you consider as an arbitrary cutoff for expression for low and high?
Excellent question, and we'll get into that a little bit better because I will cut to the chase here and highlight one of the challenges and reasons that every biologist needs to be able to dive into this data. There is no arbitrarily definable metric for assessing what level of expression you want to consider on versus off. When I teach this material to students, as many of you may do as well, I often ask what fraction of genes in the human genome, or the toxoplasma genome, or the plasmodium genome, or the Anopheles genome, or the Aspergillus genome is alternatively spliced. And you'll typically get back questions responses saying 10 genes, 100 genes, 1,000 genes, 50% of the genome, 5% of the genome, 2% of the genome. But it's a trick question because, as anyone who has explored RNA-seq data in detail knows, the answer is all of them, right? Every single gene in the human genome, or the Drosophila genome, or the Anopheles genome, or the Aspergillus genome, and so on, is alternatively spliced if you sequence deep enough. For the Toxoplasma genome, for example, there are approximately 6,500 annotated genes in the genome, and approximately 30,000 annotated introns, an average of about 5 introns per gene. But we have evidence of reproducible splicing intron splice sites that I'll show you in a moment from multiple independent experiments for over 3 million introns in the parasite genome. So you could despair over that and say that means we only know 2% of parasite biology. But in fact, the vast majority of those alternatively spliced variants are expressed at vanishingly low abundance. Could they be important? Perhaps under certain conditions or in certain strains. Could they, even those that are predicted to encode a non-functional product, could they be important as a dominant negative subunit? Regulatory subunit? Sure, but in the absence of biological evidence to the contrary, I certainly wouldn't want to bet my career or say a PhD student's dissertation on tracing down an alternative variant that accounts for roughly 1% or 10% of total message. And I'll give you some examples as we go along that highlight what that looks like. I hope that answers your question. And do feel free to interrupt if there are other questions that you'd like to try to address. So you may notice that the view of RNA-seq data that we're looking at now for this HGPRTase gene looks a little bit different than the view that was displayed here in the genome browser. And there are a couple of reasons for that, in part because the view which is captured here comes from the older technology of GBrowse. And JBrowse gives us a nice functionality of being able to summarize the expression data from multiple different studies on the forward and reverse strand, as conventionally indicated by the blue and red levels, and interspersed between unique tracks and non-unique tracks. So in one go, I can get a quick view from all of the samples in this particular study, covering different life cycle stages of the expression levels. And I can certainly see that for this gene and for the gene to the left of it, that the organization of these transcripts bears some relation to the actual annotation. I can see evidence supporting all of the annotated exons, but I might want to explore that. I might want to explore that a little bit further. So I'm going to turn on the intron coverage plots by selecting tracks, and I can see a vast array of plots that I might choose to display. But I can filter this to look at data that we're interested in, for example, to look at the transcriptomic evidence only. Look at this experiment we've been considering about feline enterocyte, tachyzoite, and bradyzoite stage transcriptomes, giving me 31 possibilities. And that allows me to select the tracks that I'm interested in. I'm only interested in RNA-seq data that maps uniquely to the genome, to avoid confusion about paralogous genes that have been duplicated elsewhere in the genome. And I'm only going to pick a single stage for a single stage for these gametocyte stages, arbitrarily at this particular one here. Here's the density plot that we were looking at before, but I'm actually going to look at the binding of unique mappers only, which will give me a little bit more revealing view. And I'm also interested in asking about the tachyzoite and bradyzoite tissue cyst stage parasites. So having selected those tracks, let me go close my select tracks window. And now you can see, because I selected these in maybe not the best order, let me reorganize these tracks to get the view that I would like. I will pick a tachyzoite track and drag that up to the top. Pull up the reverse strand for that. Maybe grab this version and drag it down to the bottom, and just to reflect the temporal life cycle, move the bradyzoite tracks up above. So simple dragging and dropping allows me to manipulate these tracks so that we're now getting a view of the life cycle. I'm going to do a couple of additional things here. I'm going to take my annotated track and pin that to the top of the page so that whatever scrolling I do, I can always view it in the context of the annotated gene. And I'm going to zoom out just a little bit so that we can maybe see a little more of the context from that region. All right. We'll spend quite a bit of time on this page here to be able to make sure that we understand the range of information that we can get from this. And then I will, because we only have an hour together, I will jump to a view that I pulled up previously, highlighting considerably more information that you might want to explore. So let's take a look at this gene. Now we can see RNA-seq coverage plots that look much more comparable to what we had seen in the genome browser previously. You'll recall that there were that expression levels varied by over 1,000-fold from one study to another, and over 100-fold within this particular study. But in fact, what we see for this gene is expression levels that appear roughly comparable across these life cycle stages. But I will remind you that we are following this using a log scale, relative expression levels ranging from 2 to the 0 to 2 to the 7th. If you are interested in looking at a linear scale, we can modify the view of our tracks, setting this not as a log scale, but as a linear scale, changing all of those X, and now we can see the, and now we can see more readily the much higher abundance in gametocyte stages than in the tissue cyst stages, which are the lowest in this sample, and slightly higher in tachyzoite stages. You can also see, as indicated by the black lines, that in this linear view, we've truncated the expression levels in this stage just to get everything on the page. and you could manipulate that for individual tracks as you wish by clicking on the ability to manipulate. There are reasons to look at both log and linear scales, and for our purposes today, at least for the time being, I'm going to go back to the log scale view. All right, what can we see in allowing us to assess the accuracy of this gene model? The comparison with the published gene models from 2 decades ago, and to consider how this ought to be re-annotated as the Apollo browser comes online, addressing the question that was raised earlier. Let's start at the upstream end of the gene, and because this gene is indicated in blue, I know that it reads from left to right, as opposed to the genes on the opposite strand indicated in red, which read from right to left, as indicated by the arrow here for this upstream hypothetical protein. I can see that the annotated transcriptional start is in fact just an approximation of the truth, right? It's annotated to start about here, and we could zoom in to see what that position is. But in fact, steady state transcript abundance levels from these Illumina RNA-Seq data sets show that transcripts start over a fairly wide range here of a couple hundred nucleotides. But again, I remind you that if we were to go in and look at this on a linear scale, we would see a tighter peak. Return to our log scale view. This messy transcriptional start site is characteristic of most genes in most genomes. We can see it at the gene that's upstream as well, that ramps up, and it's true at the 3' end as well as the transcript abundance ramps down. So as the first answer to your question about what constitutes an appropriate annotation, what would you as a biologist like us to do? Would you like us to annotate a single transcriptional start site, stop site in this case, or 200 transcriptional stop sites across the range as this ramps down? Clearly, we wouldn't want the latter, but it's important as a biologist to be aware. For example, if you were trying to design PCR primers that would capture the non-coding sequence, that steady state transcript abundance doesn't end at one particular site. Moving to the 3' end, we can see that transcript abundance ramps down in this gene in Toxoplasma as well, approximately at the level indicated by the annotation. Let's take the most abundant of these transcripts, look at it on a linear scale, and we can see that indeed this falls off pretty quickly over a range of about 100 nucleotides, roughly centered on the transcriptional the annotation, which is derived from that pre-genome publication that I showed you earlier. So the annotation of the 3' UTR here looks pretty good. However, when we look at it on a log scale, we can see that there is that a substantial number of transcripts, far fewer, right? So we have mousing over shows us the abundance level. There are 1,000, the FPKM values are about 1,000 here and 10 here. So 1% of its total message, but a distinctive 1%, shows a longer transcript. Should we annotate this as an alternative transcript with an alternative 3' termination site? That's really a judgment call, and the annotators in this case have chosen not to do so. One might wish to do so, but again, I'll remind you that if you were to annotate this with 1% of the total message as an alternative transcript, maybe you would want to annotate all of these possible start sites up here that have higher abundance as possible start sites as well. Ultimately, there's no substitute for being able to dive into the raw data to see the experimental support or lack thereof. for the studies that the questions that you're interested in looking at. What about the annotated introns? Here we can see we're on much stronger grounds. That the drop-off for these introns is much tighter. There is virtually no transcript abundance, more than 1,000-fold difference between the transcript abundance in these exons and the reads that were acquired from introns. A very tight, specific start to those genes. But once again, if we want to look at that in greater detail, we can zoom in to take a look at the actual mapping to those transcripts. And as we dive in, we see even a little bit of sloppiness on the splice junctions for this exon that's shown that's shown here. And if I were to zoom in further and turn on the sequence information, what you would see here is a very small level of splicing that leads to the addition to either a single nucleotide change, which would cause a frame shift in the transcript, or a 3 nucleotide change that would introduce an extra amino acid, or a 6 nucleotide change that would introduce yet another amino acid. These are alternatively spliced products present at vanishingly low abundance, less than 5% of total message. And to give a precise answer to the question that was before, my personal cutoff for what should be annotated and what shouldn't, in the absence of additional biological evidence, is transcript abundance that's at least 20% of the maximum. And we'll take a look at that a little bit. We'll take a look at that a little bit later on. So we can see that there's a lot of variability in the RNA-seq data, but all of these issues that we've talked about, the variable transcriptional start sites, the variable transcriptional stop sites, the precision in intron excision are all borderline cases, nothing that we would want to annotate. What about the demonstrated and validated alternative splice variant that leads to the skipping of exon 3? As we go back to look at this, we can see that the abundance of exon 3 is substantially lower than the abundance of the surrounding exons 2 and 4. In this particular sample, about 1/4 of the total, which corresponds nicely with the published analysis indicating that depending on the strain, anywhere between 20% and 50% of the protein, it consists of isoform 2. In this gametocyte stage, much higher expression, but once again, 20%. The steady state abundance of transcripts that map to this alternatively spliced exon account for only 20% of the total. We can extract that information from the reads which span these intron boundaries, and we've seen that already as extracted on the gene page when we looked at the very top, looked at this evidence for RNA-seq introns. And so I'm going to go back to turn on the track that allows us to view that information. I'll clear the filters that I'm interested in just to make sure that I'm not adding one filter on top of another. This does nothing to the tracks that are still on the page. We can see over on the far right-hand side, you can see those tracks haven't changed. I'm going to look at transcriptomic data, but in this case, I'm interested in the predicted intron junctions, not the coverage plots or multi-density plots. And while I could turn on the tracks that indicate the evidence for any of these experiments, such as the ones that we just looked at, I'm going to look instead at the evidence for introns that has been compiled from all of the available RNA-seq data, either in a refined view. Our best case assessment of what ought to be annotated, or a more expansive view, including many more, although not all, of the data from all of the RNA-Seq tracks. We'll turn that back, identify these evidence tracks, and maybe drag them up where we can see them a little bit better at the top of the page. And one more. Right. Now let's look at what the information is that we can see. Here's the purine phosphoribosyl transferase gene that we've been considering. And when we look at an inclusive view of the evidence, we can say that beyond the 1, 2, 3, 4, 5 introns that are annotated, we can see these symbols that indicate evidence from intron spanning reads that correspond precisely to the intron boundaries. If you'd like to confirm that they are precise, I'm going to modify this track to only show those introns that map the transcript annotation, and I can see that all 5, 1, 2, 3, 4, 5 of these introns are supported by RNA-Seq data, and I'll tell you in a moment what the index mean, but we can immediately see that they differ in the thickness of their lines, and that difference in the thickness of the lines corresponds to the abundance of the evidence. Let's look first at the excision of intron 4 here, the thickest of these lines, the most consistently excised in all of the experiments that we're looking at. And by clicking on that, I pull up a lot of information that's useful to be able to examine. I can see, for example, the location of the intron, the number of intron spanning reads. So this is 31,489 independent pieces of evidence in support of this intron, a wealth of support vastly greater than we could have ever anticipated a generation ago when we sequenced, I don't know, 10 or 20 cDNAs that corresponded to this transcript and led to the identification of alternatively spliced isoforms. This number, the ISR per million, is a number of intron spanning reads per million intron spanning reads, and it is designed to provide a comparison with the transcript abundance, the FPKM values, allowing us to assess whether the number of intron spanning reads that we see from the RNA-Seq data is consistent with the transcript abundance levels from that map data. I can see, looking down below, that this particular intron is the most abundant intron observed in this gene. It is most abundant in the experiment that we're looking at early gametocyte stages of differentiation. So not even the track that we're looking at. It's expressed even more abundantly at a different stage, and in the most abundant in the sample for which this intron is most in evidence, its ratio with intron spanning reads is very close to the expected levels. The expression is high at 1,000-fold, but the ratio is very close to 1. So we have every confidence that in this case, far less than 10% of transcripts are failed to excise this transcript, and we can see the same information for the many different experiments that are for which data is available, allowing us to ask, for example, whether there are differences in expression in different life cycle stages, in different experimental treatments, in different strains that have been identified. So these are the 5 annotated introns, but we can see that while intron 1 is efficiently excised, intron 2 is excised far less is observed far less frequently. Only about 20% of the total number of times. The same is true for intron 3, about 24%, and 4 and 5 are both abundantly expressed. And this, of course, would be what we would expect from the annotated view, in that exon 3 is only sometimes observed. In some cases, introns 2 and 3 are excised together. That is not one of the annotated transcripts because, of course, it hasn't been, that is not reflected in the annotation of this gene. But if we return to looking at all of the introns, we can see a vast wealth of introns that are identified associated with this gene, sometimes spanning this gene, sometimes mapping in strand-specific RNA-seq data to the opposite strand transcripts, low levels of abundance of introns in areas that aren't annotated as having genes at all. But if we were to click on this to look at the abundance of this particular transcript, that we can see that while there were tens of thousands of introns supporting the annotated lines of evidence supporting the annotated introns, there are much lower levels of expression for these other unannotated genes. The track up above, which you can't see easily here, I'll pull it down so you can see the name. Let's see. The refined track here provides our assessment based on standard cutoffs provided by curators as to introns that you might want to actually consider. So I'm going to get rid of the broader view here, leaving me with only the refined view, which we can compare with the annotated versions here. So this allows us to see that in addition to the annotated introns, 1, 2, 3, 4, 5, there's really only one additional intron that we might want to consider, and that corresponds precisely to the isoform 1 intron that includes both introns 2 and 3 and leaves out the intermediate level, it leaves out exon 3. Finally, let's turn back to this question of what we are to make of the apparent read-through of the first intron in non-coding sequence. We can see that in the RNA-seq view as well, that while there are virtually no reads that map to the fully excised introns, there is what appears to be a substantial amount of transcript abundance here within intron 1, predicted to be a non-coding sequence from the 5' UTR. But rather than being a relatively uniform level of expression, what we see is a gradual ramping up that looks something, reading from left to right, that looks something like the promoter. And in fact, this suggests to us, as we now know from experimental evidence, that our assessment from those few cDNA sequences sequenced many years ago, that we interpreted this wrongly. That in fact, in addition to the transcript start site or transcription start sites up here at the 5' end, there are alternative transcriptional start sites that initiate within the first intron, which of course makes it impossible to excise that first intron. And perhaps what we should be doing is annotating a transcriptional start here, here, and also here. These are judgment calls for annotators or judgment calls for you as biologists working in the lab, because the confidence of the annotator in what ought to be functionally annotated will depend on the biological evidence that is supporting that. Now, there are many, many additional tracks that you might want to turn on to take a look at, similar to those that were shown here in a previous version of the tutorial. And because we are running short of time, I'm going to jump to a view that I ran last night looking very similar to what we have explored previously, but I have added in here some of the additional information just to illustrate to you what is possible. I've included versions of the annotation from an automated gene finder, which recognizes all of these, and from manual curation, which has yet to make its way into the official GenBank record, which thankfully properly recognizes this exon skip polymorphism. I've turned on a series of tracks using the density view to look at 29 different strains spanning the diversity of Toxoplasma genomes identified from hundreds of sequences around the world. And we can see that to a first approximation, there appear to be no dramatic strain-specific differences in the gene models. We could go in and take a look at these here, which seem to show some possible differences if we wanted to look at possible alternative isoforms in different strains. I've turned on coding potential information to see that while there are many polymorphisms associated with the gene, the majority of them are associated with non-coding sequences in introns, for example, or in the 5' UTR. There are only two non-synonymous, there are only two coding sequence trons as indicated in blue, both of them in this alternatively skipped exon, one a non-coding polymorphism, one a coding polymorphism, which leads to an amino acid change, an amino acid change from an alanine to a glutamine in the minor allele, which accounts for only 2% of the sequences identified to date. I can see that there's proteomics data in support of this protein. Interestingly, most abundantly for the mature part of the protein where the PFAM domains are identified, but we do see experimental evidence at the protein level for this alternatively spliced, alternatively included exon, and including proteomics data, which supports the annotated gene models showing the introns that are indicated here. We can see chromatin mark data from ChIP studies and ChIP-seq studies that look at the region, for example, of the promoter, allowing us, I should be just using the mouse here to scroll down, allowing us to look at promoter binding studies, and from ChIPs and from ChIP-seq studies as well. And we could turn on additional tracks looking at comparisons with other orthologs in different species. We might, for example, want to ask whether the well-aligned sequences for other parasite isolates, looking at the comparative gene models, for example, in other look at in Neospora or in Sarcocystis or in other species, we might want to ask whether RNA-seq data for those strains is available and supports the alternative splicing of the gene, even though we can see a very different exon structure, for example, in Sarcocystis. So there's a vast range of information, and let me close by skipping over my examples from FungiDB. Apologies to the Aspergillus researchers out there. But take a look at genes in insect species, which of course are classically characterized by extensive alternative transcript processing. Alternative transcription was discovered very early on in Drosophila and is much more abundant than it is in protozoan genomes like that of Toxoplasma. I've looked at one particular gene here, annotated as a histone deacetylase, and with two different isoforms in vector base. As you know, the Bioinformatics Resource Center project is combining vector-based FungiDB and parasite genomes together. This is a first view of the of vector-based within the same infrastructure. You'll notice some consistent design. All of the information from vector-based currently has been incorporated in the platform that we've been exploring today, allowing us to look, for example, at this particular gene and its alternative exon coverage. And if we wanted to look at JBrowse to view the evidence supporting that, we can see, for example, that for this particular gene, we see across a range of sexual differentiation in anopheles, many different studies that we could dive in and look at, support for all of this evidence, but different stages seem to use or exclude this exon here, and we could look into those to see what the case is for that particular gene and evaluate other evidence as well. This RNA binding protein, for example, seems to be reasonably well annotated, but in certain strains, it looks like there's a very abundant longer 3' UTR, or alternatively another annotated gene that is here. Scrolling down further, we can see other evidence for other alternative exon genes here, and we can look at the strains in which those are used. We might want to explore whether there is indeed a third transcript that includes both of these exons. Same thing for this transcript, and so on. So with that, let me conclude by just reiterating that I encourage any of you to contact us by the link that is on the top of this page, either from the current website or the soon-to-be released new and improved merged ViewPathDB launching platform. If you have other questions that you would like to ask, or whether you would like us to work with you via via email or a private GoToMeeting or a Zoom call with you, with your lab group, with a broader team, and so on. And I encourage you to come back for the subsequent webinars in this series, including one on the Apollo platform that will show you how you can promote these alternative gene models to be represented in the annotation that's viewed by others. Let me turn things back over to you, Omar, for any questions or any last-minute comments you may want to raise in wrapping up our discussion today.
Great. Thank you, David. This was a very nice webinar, which, again, as a reminder, it has been recorded. And so we will share that link with everyone, and you'll be able to view it or share it with your colleagues as well. We had a number of questions, which I believe we've answered all of them by text, by typing them in there. So others can review those questions and answers. And if you have more questions, we'll stay on for a few more minutes and answer them as they come up. One general question that came up a few times was asking about data that's not available in the resources yet. So we encourage and we love to hear from our community about data sets that are out there that should be included. And to do that, just click on the Contact Us link, as David mentioned, and let us know about the data set, the publication, or if you have the link to the bio project, or any information you have, just to start the conversation so we can keep track of it. The team here continuously looks at the literature, and we attend meetings and conferences, and we try and find out about data sets that should be included. But of course, we're not perfect. We miss ones, and we rely on you to let us know about data sets that are important to the community that should be loaded. Sometimes we get questions about data sets that are on genomes that are not part of the project, and I would suggest that feel free to let us know about these data sets. It is not 100%, depending on the type of data and the type of genomes, we may or may not be able to include those. But the conversation can start by just letting us know about the data set, and we can carry on from there. So let's double check here and see if there are any other questions, or anybody else on the call from the outreach team. Are there any other questions that should be answered right now?
Nope.
Okay, So the other thing that I wanted to, obviously I'm showing you here on the website is just the results of people who are attending this webinar, at least the 136 participants who voted. If you still want to vote on this and let us know which resource you use, feel free to use the link that's shown up here, or you can scan the QR code that's shown on the screen here. I'll leave it up for maybe 10 more seconds because I want to show you another link that I would encourage you to use as well.
The link is in the chat too.
Great, thank you, Sam. Yeah. So, and you can feel free to answer that afterwards as well. I'll keep that poll open for a little bit longer after the webinar. So the last thing I want to show you is another link. So this is a webinar survey. So you will get this link in your post-webinar email, which will go out either later today or tomorrow. But if you get a chance right now to just scan this QR code or type this URL, I'll put it in the chat window as well, just so it's there as well. Please do so right now. I think we know from various analyses that answering a survey right after an event is always better because things are fresh in your mind. So let us know, and it's important that we get a good response rate. The answers you give us in these surveys are really important for us to shape future webinars and also allow us to gauge your interest and things we can change about the webinars to make them even better. Great, so I don't think there are any more questions coming in. So thank you everybody for joining this webinar. Feel free to sign up for future webinars. Next week it'll be on search strategies. Learn how to develop your own in silico experiment. Thank you everybody for joining. Thanks David again for a great webinar, and see you all online soon. Bye-bye.